Today, we're going on one of the most incredible journeys imaginable. Not to some distant star, but into the space between our own ears. A project between Harvard and Google has just given us a map of the human brain like we've never seen before, and it really makes you wonder. You know, it's a wild thought, right? We've got these unbelievable telescopes that can see to the edge of the universe. But the thing sitting right inside our own skull is still, in many ways, a complete mystery. And the reason for that all comes down to scale. The real action in the brain, the stuff that makes us, happens at a level so tiny it's almost impossible to see. I mean, sure, we've all seen brain scans like MRIs, and they are amazing. They show us which big neighborhoods of the brain light up when we're happy or solving a puzzle. But what they can't do is show us the actual streets and houses. They can't see the individual wires, the synapses, where all the information is actually passed along. And that's where the code of consciousness is written. To actually read that code, you have to zoom in. I mean, way in. We're talking nanoscale. We need a map that shows every last wire and every single connection. And for that, you need a totally different kind of tool, an electron microscope, which lets us see things that are smaller than a wavelength of light. And that brings us to the HO1 project. This was this massive effort to finally create that map. A team from Harvard and Google got their hands on a tiny piece of human brain tissue from the temporal cortex, and they set out to map every single thing inside of it. And when I say tiny, you won't believe how small it was. The whole sample was just 1 cubic millimeter. That is about the size of a single grain of sand. Now, it came from a patient who was having surgery for epilepsy, which is an important detail, but pathologists confirmed the tissue itself was structurally normal, which means this is the clearest, most detailed look we have ever had at our own wiring. Okay, so what's inside that grain of sand? Well, for starters, the team found over 57,000 individual cells. You've got your neurons, your glial support cells, blood vessels, the whole shebang. It's like a whole city packed into this microscopic space. Just let that sink in for a second. But here's the number that just blows my mind. The synapses, the connections. Inside that 1 cubic millimeter, they counted nearly 150 million of them. These are the little junctions that make the whole circuit work. 150 million connections in a piece of brain you could barely even see. So, what kind of hard drive do you need to store a map of a single grain of sand's worth of brain? Well, it turns out you need 1.4 petabytes. To give you some perspective, 1 petabyte is 1,000 terabytes. This data set 150 years, all for that one tiny speck of tissue. So the big question is, how on earth did they do it? I mean, creating this digital universe from a grain of sand was an unbelievable challenge. It took a mix of super precise biology, powerful microscopes, and just a massive amount of computing power. It was like trying to solve a puzzle with trillions of microscopic pieces. Here's the basic idea of how they pulled off this incredible feat. First, they took that tiny sample and sliced it into more than 5,000 slivers, each one thinner than a wavelength of light. Then a custom microscope took pictures of every single slice, creating millions of little image tiles. After that, some seriously powerful software had to stitch all those millions of images together into one solid 3D block. And then came the really amazing part. They unleashed AI algorithms to trace every single cell through that entire digital maze. And this slide just shows you how tricky that alignment step was. See the images on the left? That's the raw data. It's all shaky and misaligned, like a messy stack of photos. But on the right, after the software works its magic, you get this perfectly smooth, continuous block of tissue. Now just imagine doing that for 196 million pictures to build that final 1.4 petabyte model. It's just incredible. Now this is where the AI becomes the hero of the story. You see, trying to trace 57,000 cells by hand through thousands of images would be, well, it would be impossible. It would take centuries. So they used this clever AI called a flood-filling network. You can see it working here. The black and white image is the raw microscope data. And the colored parts are where the AI has automatically identified and filled in each cell, creating a perfect 3D model of everything inside. Okay, so building the map was a technological marvel. But the real excitement, the reason they did all this, is what they found inside. This map is so new and so detailed, it's already rewriting textbooks and showing us things we've never, ever seen before. Let's check out some of the highlights. So first off, just taking a basic count head of what's in there led to some big surprises. For one thing, the brain support cells, the glia, actually outnumber the neurons 2 to 1. And get this, the most common type of cell wasn't a neuron at all, but something called an oligodendroglade, which makes the insulation for the brain's wiring. Oh, and they also found a bunch of weird blobs that they couldn't identify at all. But then things got even weirder. Deep down in a part of the cortex called layer 6, they found these bizarre neurons. Take a look at the image here. They call them compass neurons. See how they each have one really big branch that points in one of 2 opposite directions? They form these perfectly mirrored groups. What are they for? Right now, nobody has a clue. This quote is pulled straight from the scientific paper, and it hints at what might be the most profound discovery of all. The scientists started noticing that some of the connections in the brain didn't look random at all. They looked like they were there on purpose. And this is exactly what they meant. So most neurons connect to each other with maybe 1 or 2 synapses. It's a pretty weak link. But the team found these rare, incredibly powerful connections. Check out the image here. You can see a single axon reaching out and making 53 separate connections to one single partner neuron. The odds of that happening by chance are basically 0. It suggests the brain isn't just a random web, it's deliberately forging these super connections between specific partners. So after years and years of work, this first incredible exploration is done. But here's the best part. This isn't the end of the story. It's really just the beginning. Because the team behind this map didn't just publish their findings and call it a day. They did something much, much bigger. They've made the entire thing, all 1.4 petabytes of it, totally free and available for anyone in the world to explore online. They even built tools so you can fly through it yourself. This isn't just a research paper, it's a new atlas for neuroscience. It's a gift to science that's probably going to lead to discoveries for decades. And that just leaves us with this final, kind of humbling thought. This entire journey, all of these wild discoveries, came from a piece of our brain no bigger than a pinhead. So it just makes you wonder, what other beautiful, bizarre, and unimaginable secrets are hiding in the billions of other millimeters that make up the rest of the human brain? All right, today we're diving into one of the biggest, most ambitious challenges in all of science. It's a question that smashes together biology, computer science, and honestly, a little bit of philosophy. It seems like such a simple question, doesn't it? But the answer is, well, it's unbelievably complex. To even think about simulating a brain, you first need a perfect blueprint. And then you've got to have a way to test if your simulation actually, acts like the real thing. So let's see how scientists are tackling both of these massive problems. So here's the game plan. First, we'll dig into the incredible, painstaking effort to map the brain's wiring. Then we'll see if we can use those maps to actually predict what the brain does. We'll check the report card. How well are we doing right now? And finally, we'll look ahead. What's next on this absolutely monumental quest? Okay, part 1, the blueprint, or connectomics. Every big construction project, right? It needs a blueprint. For the brain, that special blueprint is called a connectome. It's the full map of all its crazy, intricate wiring. Think of a connectome as the ultimate circuit diagram. It's a map that doesn't just show you every single neuron, but, and this is the important part, every single connection, every synapse between them. This is the physical hardware that makes all our thoughts and actions possible. And making one of these maps, wow, it is a truly mind-boggling task. First, they take brain tissue and slice it into thousands of layers, each one thinner than a human hair. They image every single slice with an electron microscope. Then, powerful computers stitch all those flat images back together into a 3D model. And after all that, human proofreaders have to spend literally tens of thousands of hours just correcting the mistakes the computers made. It's a huge effort. All of that incredible work led to a massive breakthrough you see here, the HemiBrain project. This is a nearly complete wiring diagram for a big chunk of a fruit fly's brain. It's a huge landmark, our first really detailed blueprint of a brain this complex. And the scale is just staggering. We're talking about almost 25,000 neurons and 20 million connections between them, all mapped out. This gives us a look we've never had before into the circuits that control everything a fly does, from flying to finding food. But let's be real, a fruit fly brain, as cool as it is, isn't a human brain. So what happens when you try to use these same techniques on a piece of our own noggin? Well, this is what happens. This is the H01 dataset. It is a map of just 1 cubic millimeter of the human brain. That's a tiny sample, smaller than the head of a pin, that was taken from the cortex of a patient during a surgery. And here is where things get absolutely wild. This tiny, tiny piece of human tissue has an estimated 150 million synapses. That is more than 7 times the number of connections in the entire fruit fly hemibrain we just saw. The data for just this 1 cubic millimeter takes up 1.4 petabytes. That's like a million gigabytes. It's just an insane amount of information. So we're getting these blueprints, these amazingly detailed static maps. But a map of all the streets in a city doesn't tell you anything about the flow of traffic, right? To get how the brain works, you have to see it in motion. Which brings us to part 2, the test. This is where we try to go from that static map to a living, breathing, dynamic simulation. And the ultimate test of that is prediction. Yeah, this really puts the challenge into perspective. If the connectome is the brain's hardware, the physical wiring, then a good simulation has to predict the software. It has to predict the actual patterns of electricity, the neural firings that flash across that hardware from one moment to the next. So for this test, scientists needed another perfect subject. And they found it in the larval zebrafish. Because these little guys are transparent, we can actually use powerful microscopes to watch and record the activity of pretty much every single one of its 70,000 or so neurons, all at the same time, while the fish is alive and kicking. It's the only vertebrate animal where we can do this right now. This incredible ability led to something called the Zap Bench Challenge. You can think of it like a weather forecasting problem, but for the brain. The researchers take a few seconds of real, recorded brain activity, feed it into a computer model, and then the model's job is to predict what all 70,000 of those neurons are going to do for the next 30 seconds. Part 3, the verdict. So, this is the moment of truth. How did the models do? Can our current tech actually predict what a living brain is going to do next? And this is so critical because, as the creators of ZapBench themselves put it, prediction isn't just a cool trick. It's a fundamental test. If you can't predict what a system is going to do, you can't really claim that you understand. And here, you can see the results for yourself. On the left, that's the ground truth, the real, beautiful, shimmering patterns of activity that were actually recorded from the fish. On the right, that's the prediction from one of the best computer models. And you can see it gets the general vibe, right? The right areas are lighting up, but those fine grains, specific little flickers of light, they're just not quite the same. This chart really nails it down. If you just make a naive guess, like assuming nothing changes, your accuracy is way down here. The machine learning models are much, much better, no question. But they're still a very, very long way from a perfect prediction up here. So the takeaway is pretty clear. Our ability to predict what an entire brain is doing is promising. It's definitely better than just guessing, but we are nowhere near a perfect simulation. We're capturing something real, but there's still a massive gap. Which brings us to our last section, the future of simulation. So what's holding us back? What are the next big hurdles on this epic scientific quest? Well, the list of challenges is pretty long. For starters, our data itself isn't perfect. Those connectome blueprints still have tiny errors. And our imaging, while amazing, is still too slow to capture the true millisecond speed of neurons. But then there's a whole bunch of biology we're just beginning to understand. Things like glial cells, which, by the way, actually outnumber neurons in the human brain, or other ways neurons talk to each other. Our models just don't account for that stuff yet. Look, what we're seeing from projects like the Hemibrain, H01, and ZapBench is absolutely groundbreaking science. We have our first real blueprints and our first true predictive tests. But it's so important to see these for what they are. They aren't the end of the journey. They're the very, very first steps. The starting gun has just fired. And all of this leaves us with one final, really deep question. Let's just say decades from now, we solve all of these problems if we finally build a model that can predict every single neuron's firing perfectly. Have we actually simulated a mind? Or have we just built the world's most complicated puppet? That's the question that sits right at the heart of this entire quest. Thanks for joining me.